The first biomolecule that is present on our list, it is the carbohydrates. Carbohydrates are considered as the simplest form of biomolecules since they are only possessing carbon, hydrogen, and oxygen on their backbones. Because these are the simplest materials and simplest atoms that is actually included and incorporated on the different types of biomolecules that we are going to discuss in this discussion. Carbohydrates are actually considered as polyhydroxyaldehyde and polyhydroxyketones. They are considered as polyhydroxyaldehydes or polyhydroxyketones because of the functional groups that is present in every single one of them. Because carbohydrates are possessing ketone and aldehyde functional groups. And these functional groups are actually very vital on the creation of more complex materials that's related to carbohydrate metabolism. After the presence of the functional groups known as aldehydes and ketones, as mentioned earlier, they are simplest in forms and serves as fuel and energy storage. We can actually observe here the different types of monosaccharides. Monosaccharides, these are the monomers of carbohydrates. And you can observe here that monosaccharides, once again, as mentioned on your module, are the simplest form of sugars, and their carbon backbones ranges from 3 to 7. But what you actually have here are the most common types of monosaccharides. Namely, these are 6-carbon monosaccharides known as hexose. We're going to consider the different monosaccharides. We have here the glucose, galactose, and fructose. These are the most common types of 6-carbon compound sugars. As you can observe, glucose and galactose, they're actually possessing aldehyde functional groups, while the fructose, it is possessing a ketone functional group. This is the ketone, then these are the aldehyde functional group. Also, you're actually going to observe that when it comes to glucose and galactose, they're actually possessing here a part known as a stereoisomer or epimer. Or, it is just a mirror image of a portion of a certain type of molecule. Now, if we're going to have a deeper discussion regarding this, once again, we have the presence of the concept of isomer. Isomer, these are the different chemicals which are possessing same chemical formula but different chemical structure. So for instance, we have the presence of glucose, galactose, and fructose. All of them are isomers of hexose or 6-carbon sugars. They are isomers of hexose, while epimers or stereoisomers, they are known now as the different types of molecules that are possessing different configuration or reflection or mirror image. So once again, galactose and glucose are epimers of each other, while isomer, the fructose, glucose, and galactose are all isomers of the hexose. Okay? So remember those things whenever we are talking about isomers and epimers. After the monomers, which are the monosaccharides, let's now proceed to the simple polymers, which are known as the disaccharides. Now, once again, the discussions that is present here are just minimal, while majority of the discussions are already present on your modules. I'm just having short rundowns of the different things on the PowerPoint presentation. Then, later on, the important details will only be discussed every single one of them. Just like this case. You can observe here that whenever there is the presence of dehydration reaction between a glucose and a glucose, which will now create a maltose. We give off hydroxide, then hydrogen ion to create water. Then, there will be a glycosidic linkage or the linkage that is present between two monosaccharides. Then, it will now create maltose. Then, in the case of the creation of sucrose, it is just a combination between glucose and the fructose. Glucose will now give off hydroxide, while hydrogen ion will be removed from the fructose. Then also, once again, they will have a glycosidic linkage, which is now the connection between the glucose and the fructose to create a sucrose. These are actually just the different mechanisms on how disaccharides are formed. And you can also see on page number 5 of your module, we have here the different disaccharides, namely the maltose, lactose, and sucrose. Maltose, it is a combination of glucose and glucose. Lactose is the combination of galactose and glucose. And lastly, fructose and glucose, when they combine, will now create sucrose. Now, after the simple disaccharides, let's now proceed with the complex polysaccharides. The complex polysaccharides can be determined as starch, glycogen, and agar-agar, and carrageenan. While in the case of structural polysaccharides, we have here the presence of cellulose, pectin, and chitin. So these are just some of the different polysaccharides that are present. And once again, the majority of the discussions are present on your module so that the important details will only be included in our video presentation and the bulk of the discussion will be present on the module. And also there is an introductory video regarding biomolecules that will be flashed on this discussion. Let's proceed to the next part. Second biomolecule, it's now known as the lipids. Now, in the case of lipids, unifying features of lipids is having little or no affinity to water, or technically, they are hydrophobic because they consist mostly of hydrocarbons which form non-polar covalent bonds. Whenever we're talking about hydrocarbons, these are just carbons which are possessing saturated hydrogen on their backbones. As you can observe here, in the case of fats, they are known as the different types of hydrocarbons because these are just hydrogens which are attached to the carbon backbones. And now, also as you can observe, whenever we are talking about the hydration or dehydration reaction to create now triacylglycerol or the polymer that is present among lipids or fats. We have here the dehydration reaction in which hydrogen ion will be removed from the glycerol backbone. Then, on the fatty acid, it will actually remove hydroxide. Okay, so that is actually how or the polymers of fats known as the triacylglycerol are created. The polymer of fats, it is actually composed of the glycerol, which is a 3-carbon backbone, and the fatty acids, which are hydrocarbons. And these hydrocarbons will now connect with the glycerol to create the first type of polymer present among lipids, which is known as the triacylglycerol, or a fat molecule possessing 3 fatty acids, and in which we can actually classify fats based on the different types of fatty acids present on every single one of them. We have the presence of saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are possessing single bonds of carbon backbone linkage. There are no bonds or double bonds nor triple bonds. They are just saturated with single bonds. Then, the next part, we have the unsaturated fat, which is now possessing fatty acids which are possessing double bonds or triple bonds. Saturated fatty acids are usually solid in room temperature, while unsaturated fatty acids are normally liquid in room temperature because of the bend that is present on every single one of them. And these bends are also known as the omega. So if you're actually going to hear the omega 1, omega 3, omega 6, these are actually the different double bonds that is present on those fats. Meaning, those fats are liquid in room temperature and those fats are healthier than saturated fats. Other than the presence of the triacylglycerol, which is an example of a polymer of this fatty acids or of these lipids, we also have the presence of the phospholipid. It is actually a type of a polymer of fats, which is now possessing the presence of a phosphate group. That is why it is known as phospholipid. Phospholipid from the phosphate that is present here, and the phosphate will simply be attached on the glycerol backbone. Remember that all fatty acids are possessing glycerol backbones. And after the glycerol backbones, phosphate will be attached on one of its backbone, while the other two of the backbones of the glycerol, that will now be the point in which the fatty acids will be placed. These fatty acids can either be saturated or unsaturated. It depends upon what type of fatty acid will be needed and will attach on that glycerol. And as you can observe here, the phospholipid will actually create two parts, a hydrophilic head and a hydrophobic tail. From the name itself, whenever we're talking about hydrophilic, it is affiliated with water or it is water-loving. It has an interaction with water. While hydrophobic, or the tails, they have no affinity to water or they do not like water. They will not mix with water. As you can observe here, in the case of our cells, they're actually possessing here the phospholipids. And on the surface that is present on the water and even in the inside of this membrane, the hydrophilic head will now be either on the innermost or on the outermost layer of the plasma membrane or of this membrane, while the hydrophilic tail will be present inside. They will just curl up inside. They will just be present here at the center, while the hydrophilic head, it will have interaction with water. The hydrophobic tail will not have any interaction with water, as simple as that. Then, we have here a list of the different types of fatty acids based on their backbones. And also on your module, you have there the discussion of the different special types of fatty acids other than phospholipid. You also have there the triacylglycerol, the sphingolipids, the waxes, and the steroids. Majority of the discussion there are actually self-explanatory, but I would like to focus on this part when it comes to our discussion, since this is actually important to every single one of us. Now, we have here the concept of trans fat. Trans fat is actually present on a lot of food products that we are consuming, especially fast food products. For instance, on donuts, on lard, on burger, on French fries, there are actually different types of trans fat. And if you're going to check the nutritional label or content of the food products that you will consume, there's a possibility that there are some percentage of it that is possessing trans fat, which is bad for your health. Why? Trans fat are the ones that is responsible for raising bad cholesterol levels. The reason why they are known as trans fat because they are possessing trans attachment of the double bond to another carbon backbone, or illegal attachment in the simplest terms. You're going to study on your chemistry class what is the difference between cis, what's the difference between trans type of attachments. In the case of trans fat, the food examples that we have here are cookies, donuts, cakes, and fries. They actually raise bad cholesterol levels on our body. Saturated fat is also bad on our body since they are possessing single bonds only. While in the case of unsaturated fatty acids, they are much better in our body than saturated fatty acids and trans fat on our diet. So remember, if you want to stay healthy, be healthy, do not forget to use the unsaturated fatty acids since they are better than saturated fatty acids and trans fat. And remember, fat is still important in our diet. You should not remove fats, you should not remove oils because majority of our cells are utilizing fats. It's very vital for every single one of us. Other than the fats and the lipids, let's now proceed with the proteins. Now, in the case of the proteins, they're actually the one responsible for the 50% of the dry mass of most cells. And due to structural diversity, their functions are also diverse. And as mentioned on your module, they are possessing the following functions: enzyme catalysis, defense, transport, support, motion, regulation, and storage. And the monomer of proteins are known as the amino acids. Amino acids are organic molecules with carboxyl and amino group. The amino group, it is the combination of nitrogen and attachment to a carbon backbone, then connection of two hydrogen atoms. While in the case of a carboxyl group, it is now the attachment of a carbon to another carbon, double bonded with oxygen and single bonded with OH. And this is actually the peculiar thing regarding all amino acids. Amino acids are actually possessing here the side groups or the R groups. The side chain or the R groups are responsible for determining the different types of amino acids present in this certain type of cell or a certain type of mechanism. Okay, remember, amino acids differ in their properties due to different side chains. And what are those different types of amino acids? Do not worry, I'm going to post this PowerPoint presentation on our module and on our Google Classroom for you to be guided regarding what are the different things that you should remember and what are the different things that are simply omitted on the discussion. You can observe here that these are the different amino acids and some of them might ring a bell to every single one of you. We have glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Also, we have serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Also, we have aspartic acid, glutamic acid, lysine, arginine, and histidine. And as you can observe, amino acids are not only acids. Some amino acids are actually basic in nature. Some of these amino acids are actually neutral in nature, and some of them are acidic in nature. Not all amino acids are acidic. Remember that. Now, as you can observe here, this is now the synthesis of amino acids. You can observe here that in the case of the amino group, it will remove hydrogen ions, while hydroxide will be removed by the carboxyl group. Remember, carboxyl group will give up hydroxide, while amino group will remove its hydrogen ions for the water to be released and to create what we call as a peptide bond. Peptide bond is the combination of two amino acids. Peptide bond is present between two amino acids. Peptide bond, if this is amino acid and another amino acid, when they combine, peptide bond is the one that is present in between the two of them. Amino acid, in between the two of them, we have the presence of the peptide bonds. Okay, so that is the case whenever we are talking about the peptide bonds and combination of one amino acid to another amino acid. And lastly, after the different proteins, let's now proceed with the nucleic acids. Now, in the case of the nucleic acids, they're actually the complex organic polymers that store and transfer genetic information within a cell. And actually, we are going to highlight the discussion regarding nucleic acid on our module number 10 or 11 probably during the second quarter, because in this discussion, we're just going to consider nucleic acid as a biomolecule. But on that discussion, we are going to consider now the nucleic acid as a very vital part of our genetics. Since I want to highlight nucleic acid on the concept of genetics, let's just have a short discussion regarding nucleic acids. Once again, majority of the bulk of our discussion regarding nucleic acids will be included on our discussion on the second quarter for genetics. So please bear with me when it comes to this part of our discussion. Now, in the case of nucleic acids, it is actually possessing basic anatomy. It is possessing a phosphate group, a sugar backbone, and a nitrogenous base. The sugar backbone that is present among nucleic acids, it is known as the pentose, which is either a deoxyribose or a ribose. The reason why it is called pentose because it is possessing 5 carbon backbones, and each carbon backbone is known as 1', 2 prime, 3 prime, 4 prime, and 5 prime. Okay, so it's very special when it comes to nucleic acids. Every single carbon backbone is termed as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So those are the different things that you should remember when it comes to the carbon backbones present on the 5 carbon sugar that is present among nucleic acids. The sugar backbone is either ribose or deoxyribose. Proceeding to the next part, the phosphate group. The phosphate group, it will just simply be attached because that will be a point of connection for the different nucleic acids for them to attach with each other. So the attachment between nucleic acids, it is actually happening among phosphate groups. And lastly, nitrogenous bases. In the case of nitrogenous bases, we have here actually 5 different types of nitrogenous bases. They are classified either as purines and pyrimidines. Purines, it is present both on DNA or the deoxyribonucleic acid and RNA, which is ribonucleic acid, which are termed as the adenine and guanine. Adenine and guanine, they are purine nitrogenous bases because they are possessing 2 rings to create now the nitrogenous base. Once again, adenine and guanine, these 2 purines are present both on DNA and RNA, while the pyrimidines, they are single-ringed nitrogenous bases, and cytosine is present both on DNA and RNA, while thymine and uracil, and if you're going to observe here in our illustration, is present either on DNA only or on RNA. Thymine is present on DNA, while uracil is present on RNA only. Okay, so remember, there are different types of nitrogenous bases. Once again, we are not yet talking about the nitrogen pairing rules. We are just talking about the basic anatomy of nucleic acids. We're going to have a separate discussion for them. But remember these parts. We have nitrogenous bases, which are categorized as either purines and pyrimidines. Purines, they are possessing 2 rings, while pyrimidines are possessing only 1 ring. Purines are adenine and guanine, while pyrimidines are cytosine, thymine, and uracil. Now, for the last part of our discussion, let's now consider here the dehydration reaction or the synthesis of a polymer of the nucleic acid. As you can observe here, on the very first nucleic acid, the first nucleotide, which is the monomer of the nucleic acids, the first nucleotide, you can actually see here that on the T' of its sugar backbone, it will actually release hydrogen ions, while hydroxide will be released by the phosphate. The role of the phosphate is to have connection between different nucleotides. It is the junction, it's the connection point. And remember, the connection is between carbon number 3 or 3', the hydrogen ion, and the hydroxide that is present on the phosphate group. Water will be released, and there will now be an attachment between a nucleotide and another nucleotide, creating a short polymer of nucleic acids. And that simply concludes our discussion when it comes to biomolecules. Once again, this is just a short rundown of the different things that we should consider, that you should consider when it comes to this discussion. Majority of the discussions are already present on your modules. So once again, thank you for listening and watching this video presentation. I am hoping that you learned a lot from this video presentation, and see you on our future consultations.